Hello everyone. Welcome to the new session of Role of Genetics in Plant Breeding. So this is the first part for the plant breeding. So introduction to plant breeding. So what is plant breeding? Plant breeding is the use of natural and artificial selection. So the breeding that occur naturally as well as artificially. Man uses artificial selection for the improvement of crops, right? So that is used to produce heritable variation, the variation that can be transferred from one generation to the next. So that is called as heritable variations. And the novel combinations of alleles in plants to identify plants with novel and useful properties. So what is the use of plant breeding? We use plant breeding for having heritable variation, the variation that can be transferred from one generation to the next generation, right? So for that we use plant breeding, and also novel combinations of alleles, right? So this gives you the recombinant or recombination recombination. So the recombinant that is matched with the, so novel combination of alleles means there is a union of or the combination of such alleles that is different from the parental one, and that gives you the new recombination, right? And identify plants with the novel useful properties. So that type of recombinant which is having a useful properties, that has to be identified, then only that plant breeding will be beneficial for us. Okay, so again, the plant breeding is the use of natural and artificial selection to produce heritable variations and novel combination of alleles in the plants, and that to identify plants with novel and useful properties. This is plant breeding. So what is the goal of plant breeding? The goals of plant breeding are to produce crop varieties that boost unique and superior traits. The plants or the crops must have superior quality or traits. Okay, so that is why the plant breeding is used to have a crop with good traits or superior traits, right? So some of the most frequently addressed traits are like biotic and abiotic stress tolerance. So there are so many stress to the plant, right? So the plant can bear that stress. So that stress can be biotic from the different organism. Like there are so many pathogens that create stress to the plant, that create stress to the crop. And every year we are having a great loss of crop due to the biotic stress as well as abiotic stress, right? So we must have a plant that is stress tolerance, that the crop that can tolerate that particular stress given by the biotic as well as the abiotic factors, right? So the plant breeding is a technique that create a crop with superior traits and that can tolerate the stress. Okay, grain or biomass yield, we always want the crop with higher yield, right? So we will choose such alleles in a crop that can increase the grain or biomass. Okay, so in the plant breeding, such alleles can be identified, and according to that, crops are also identified, and breeding or the cross has been done with the help of the breeding technique, the plant breeding technique, right? So we can obtain grains or biomass of higher yield in the crop by using plant breeding technique. Ease of processing, harvesting, milling, baking, malting, and blending. So we must have a crop with such a processing. Only the higher yield crop is not required. Later on, whatever the processing of the crop is there, that should be human friendly, right? Harvesting, milling, baking, malting, and blending. These are some of the processing technique that is used after the grain is obtained or the harvested, right? So these things are also taken in account in the plant breeding technique. And use quality characteristics such as taste or the concentration of the specific biological molecules like protein, sugar, lipids, vitamins, and fibers. So these biological molecules must be present in a good amount and a high concentration in the crop which we are obtained by the plant breeding, right? So this plant breeding technique is used for these goals. The goals of the plant breeding are biotic and abiotic stress tolerance, higher obtained of grain or the biomass yield, ease of processing, that is the harvesting, milling, baking, malting, and blending, and also the end used quality. What is the quality of the crop which we obtain, right? The concentration of the specific biomolecules must be at a greater level, that is the protein, sugar, lipid, vitamins, and the fibers. Okay, so these are certain goals of the plant breeding technique. Okay, so again, the goals of plant breeding technique we can understand from here also. Higher yield, it is used for higher yield. Improved quality, we can improve the quality of the crop by plant breeding technique. We can have the resistance to the disease and pest. Okay, resistance to the disease and pest, and we can have we can minimize our loss of crop every year by using plant breeding technique, right? Elimination of the toxic substances and the resistance to the abiotic stress. So all these are the goals of the plant breeding. We will see how it happens, right? The specific goal of the plant breeding project is highly dependent upon the market for which the product is intended. Yes. So what is the requirement, today's requirement from the crop? Okay. So according to that, the plant breeding project is started, or it choose that which of the plant breeding technique we have to follow and what are our requirement, market requirement. For example, in wheat, varieties breed to produce high levels of gluten protein are used to produce noodles and breads. So for the noodles and bread, the wheat which is used for making noodles and bread must have high gluten protein, whereas the varieties specifically selected for the low protein grains are used in the production of the pastry flours. So for making pastries, we required low protein grains. So we can see here, just for one crop, we require higher gluten protein for making noodles and bread. But at the same time, we require less gluten protein for making the pastry. So we are having two types. Our requirement is two type of wheat, right? So this is our market requirement. So according to these requirement, we can have wheat taken as an example for plant breeding technique. Okay. So this is the way. The varieties of grapes developed for the various applications such as juices, red and white wines, jams vary considerably in the chemical composition. Okay, so we require grapes for the different application, for making juices, for making wines, for making jams, right? So the plant breeders employ a variety of techniques to improve the genetic composition of the crop, and a successful strategy is dependent on the physical and physiological and hereditary characteristics of the plant. So these characteristics, like physical characteristic of the plant, physiological characteristic of the plant, and hereditary characteristic of the plant, is taken in account for the plant breeding. And plant breeding use these three types of strategy to have a different varieties of the grapes, because we require a different types of grapes for juices, different types of grapes for making red and white wines, and different types of grapes for making jams. Our requirement is different, so our grapes must be according to that. So how to prepare that grapes? So we are dependent on the plant breeding technique. We are dependent on, for this, we are dependent on the plant breeding technique. And plant breeders, what they do, they use these three strategy. That is, they are dependent on the physiological, physical, and hereditary characteristic of the plant for plant breeding. Okay. A different breeding approach are required for self-pollinating, cross-pollinating, and clonally propagated plants. So we are having different breeding approach. Self-pollinating, self-pollination, like on the same flower, the pollination is adapted. So that is self-pollination. Cross-pollination, two different flower on the different plant even. So that is cross-pollinating. And clonally propagated plant, that is all totally tissue-based culture, right? So here we are adapting different approaches for the breeding technique. So according to our requirement, okay, we choose our approach, the plant breeding approach, right? The method used by the plant breeders have developed along with the advancement of human civilization and have expanded to incorporate humanity's increased knowledge of the genetics. So we will study about the history of the plant breeding also, and there we will understand how the genetics has been progressed, how the cell biology has been progressed, and on the basis of that our breeding technique also progress, right? So important activities of plant breeding: creation of variability through hybridization, tissue culture, and mutagenesis. So these three variability technique we have adapted for the plant breeding: hybridization technique, tissue culture technique, and mutagenesis. Okay, so we can have the variability in the genetic composition of any plant which we have taken in account. Evaluation of the selected type of genotypes. Identification of the superior genotype based on the testing. So we follow the testing, and according to the testing, we take account the superior genotype. We take out the superior genotype. Seed multiplication and distribution of new varieties to the farmers for the commercial cultivation. So this is the last step, how that particular plant breeding technique result should go to the farmers. That is the commercial, following the commercial cultivation. So these are the different important activities that comes under the plant breeding. So coming to the history of the plant breeding technique. Okay, in broad sense, history of plant can be divided into four timeline, and that is pre-Mendelian era, that is before 1900, Mendelian era 1900 to 1920, post-Mendelian era, that is 1921 to 1950, and after 1950 comes the modern era, right? So The most rudimentary forms of the plant breeding likely originated during the transition of the early human societies from the hunter-gatherers groups to the agrarian communities. So, in when the human were hunters and gatherers, then also they are following the plant breeding technique. However, it also been argued that the forms of plant breeding occurred even before the human diet was based on the cultivated crops, because many plants were used in medicinal and religious capacities even during the nomadic periods of the human prehistory, when the man were nomadic, when the man were moving from place to another. On the basis of that also they are choosing their crops. They are choosing their crops based on the cultivated crop and also the medicinal and the religious capacities. Okay, so they were not using a plant breeding technique at the level of what we are doing today, but at the initial level also they were following the plant breeding technique. Pre-Mendelian plant breeding. The plant breeding has been practiced for over 10 million and arose independently in the several centers including South and Central America, Asia and the Fertile Crescent and the New Guinea. So these are the several centers, okay, where the initial plant breeding technique was used. While early farmers recognized that at least for inbreeding crops like baguette, the genetic aspects of the plant breeding were enigmatic until the relatively recent work of the Mendel. So initially they were using, but Mendel make it a full-fledged concept of the plant breeding. So Mendel was the first person who brings the initial concept of the plant breeding. So we will see how it has been done. Early plant breeders practiced a form of mass selection in which the plants of the superior phenotypes were selected and the seeds of these plants were replanted during the next season. So how they have done, suppose there is a crop land and they are having a certain types of crops. Suppose I'm taking an example of wheat, right? And they are having 3 different types of wheat here, A, B, C, right? So this can be A, this can be A, this can be B, this can be C, this can be, okay, like this. So what they saw, C is having disease resistance, A is having good yield, but causes disease frequently, right? And that B1 is having good yield as well as disease resistance. They are having both the quality, disease resistance. So according to their visualization of the crops, okay, they have chosen B1 is the best for them. Okay, so what they will do, they will cultivate it, all the types of wheat here, and they will choose the seeds of B. Okay, and they will use this for the mass selection. This is called mass selection, and they will use these seeds for the next season of growing their crop. So line developed by the early plant breeders are the term land races. So like this, they are able to take out or to select some of the seeds of the wheat or any other crop. So this is showing the superior phenotype. The B1 is showing the superior phenotype, and they can use those seeds for their planting. Locally adapted lines shaped largely through either natural selection or the farmer driven selection. So we have studied about the farmer driven selection. Natural selection is the one that nature select the best crop. Other crop will not survive, nature will select the best one. And in a particular region, these are colloquially known as the folk varieties. Okay, so they use such type of varieties. The first implementation of the controlled crosses is attributed in the 18th century German botanist, that is the Joseph Korleuter. The use of controlled pollination to make new varieties was employed with increasingly greater precision during the 19th century. So, in 19th century, starting of the 19th century, cross pollination or the controlled pollination technique was used, and particularly in maize, which received the wide attention from the early plant breeders due to its many visible phenotypes. So this controlled pollination was earlier used in 19th century for this maize crop. Theories regarding the hereditary abounded in the 19th century among both zoologists and botanists. In 1956, Louis de Vilmorin published his studies on the hereditary on beetroot and reported that progeny from two seemingly similar plants can exhibit emerging traits. Okay, so this was the earliest published, came from the hereditary background, taking beetroot as an example. De Vilmorin was thus among the first to recognize the distinction between genotype and phenotype, although so he was the first person to distinguish between the genotype and phenotype, although he did not yet formally identify these features as such. Okay. The work of these pre-Mendelian plant biologists represent that early synthesis of botany and hereditary into an emerging sub-discipline of the plant breeding. So this botany and hereditary, both these techniques, both these, even we can say the subject, were given rise to a newer discipline that is called as plant breeding. However, it was not until the work of the Mendel that the modern techniques of the quantitatively manipulating hereditary of the plant population becomes possible. So only after the theory which Mendel has given, that was used for the creation of the modern techniques of quantitatively manipulating hereditary of the plant populations. Okay. So here comes the Mendelian plant breeding. Okay, so Mendel's foundational work proved that plant breeders could predict or manipulate the genetic makeup and new varieties. So we can even manipulate. He was the first person that said that we can manipulate and predict the genetic makeup of the new varieties. Okay, so in the midst of 1800s, Gregor Mendel outlined the principles of the hereditary using the pea plant and thus provided the necessary framework for the scientific plant breeders. So his principles were the concrete, you can say, this was the starting for this newer branch of biology that is called as hereditary. Okay, or we can say the genetics. So he outlined the principles of heredity using a pea plant, and that provided the necessary framework for the scientific plant breeding techniques. So he was the first person. He mainly studied the pea plant because they had a distinguished characteristics and they were quick to grow, right? So he chosen this particular plant because it was quick to grow. And Mendel would create hybrid from these plants. So he can create a hybrid. So he can predict the result. And hybrids are the blending of the two things to make one. So Mendel would observe the 7 different traits from these plants. So what were he chosen the 7 traits, right? What was that? Flower color. And the contrasting flower colors were purple, or white. And this is all present on the pea plant. These 7 characters he chosen from the pea plant for his hybridization work. The first one was the flower color, purple and white flower, means purple pea plant and white pea plant. Okay. Flower position. What is the position of the flower on the floral axis? That is the axial or the terminal. Okay. He also taken the stem length and account for this hybridization process. That one is the short and tall. So he chosen tall pea plant and the short pea plant for his hybridization process. Seed shape, round and wrinkled. Seed color, yellow or green. Pod shape, inflated or constricted, and pod color, yellow or green. So we have studied some of these traits in the our syllabus for the botany, that is the hybridization technique or the genetics principle, right? So we are familiar with some of the traits that Mendel has chosen, that is the flower color, stem length, okay, seed shape, seed color, and pod color. So these are some of the familiar traits that we have studied for the Mendel work. Mendel 1865 paper based on the quantifying the principles of inheritance provided the basis for all modern genetics. So his paper was published, but it was sadly undiscovered until the beginning of the 20th century. For 35 years, his paper was unnoticed. And as a human understanding of inheritance and the gene action rapidly dependent, the application of these principles to the crop production was pursued with equal alacrity. So, this is the basis for the plant breeding technique. Mendel's work was the basis for the plant breeding technique, but his work was unnoticed because he was the pioneer person that uses this hybridization technique at this particular level. First person, okay? So there were no other person who was studying in this way, okay, who was doing the experiment based on the pea plant or any other type of plant, okay? So his work was undiscovered. Later, what happened? In 1900, the work of the Mendel was independently rediscovered by Hugo de Vries, 3 scientists. The first one, Hugo de Vries from Holland, Karl Correns from the Germany, and Erich Streschmark from the Austria. They came with the same result after 35 years, what the Mendel had already published. Later on, these principles were further clarified and confirmed by the several workers, and some new concept of the hereditary were discovered. So they find that the Mendel had already discovered these things which we are discovering now, 35 years back, right? Okay, so post-Mendelian work, Johnson also defined the role of environment in the inheritance of the quantitative traits, a concept that is critical to the proper execution of field experiment in the plant breeding. So he defined the role of environment in the plant breeding. Johnson. Marcus Roets. In 1933, he incorporated the use of cytoplasmic male sterility, very important point, genes into the maize breeding. This permits the controlled crossing of the plants without the need of labor intensive and the removal of male reproductive structures. Okay, so cytoplasmic male sterility, this was the technique which was used by the Marcus Roets in the plant breeding using the maize plant. The establishment of the Haber-Bosch process in 1910 to artificially produce ammonia provided plant breeders with the new goal of adapting varieties to take advantage of the abundant amount of nitrogen fertilizers. For the nitrogen fertilizer also, this technique was used by Haber-Bosch. In the later half of the 20th century, the plant breeders incorporated techniques aimed to increasing diversity and expanding the breeding process even. Following the work of the Thomas Hunt Morgan, Hermann Muller in 1910s, in 1920s, the plant breeders saw the value of physical and the chemical mutagenesis. We have studied that hybridization, controlled pollination, self-pollination or cross-pollination, and mutagenesis were the certain technique that were used for the plant breeding process. Okay, so chemical mutagenesis were also used for the production of new phenotypes. The application of statistics and the population genetics to the plant breeding was accomplished through the work of scientists such as Nikolai Vevlo and J.R. Harlan. So statistics and the population genetics now came with plant breeding. Elucidation of DNA as the genetic material by the Watson, Crick and the Wilkins, and the later deduction of the genetic code further empowered the effect of the plant breeders through increased comprehension of the genetic gene action. Now the gene also taken in account in the plant breeding technique, and this was the first elucidation that was given by Watson, Crick and Wilkins. Transfer of the genetic material between the disparate species was aided by the use of tissue culture method and later through the transgenic technologies. So we are having nowadays tissue culture technique and transgenic technologies for better plant breeding technique. Okay, so this is all about the introduction of the plant breeding. We will have some more session to understand more about the plant breeding and its application. Thank you very much.